Okay. You guys can see my screen, right?
Yes, sir.
Okay. Let me know when we can start, okay? You can wait for 5 more minutes if
Sure, sir.
required.
Good evening, Dr. Anudeep. How are you?
I'm doing great. Thank you. Thank you for having me. And how are you?
Guys, am I audible?
Yes,
Yes,
you
sir.
are.
Yeah.
I can't hear anyone. I don't know. Am I audible or not?
Yes, Bhavya, can you hear us?
Jeez. Okay, I think we shall be starting in another 2 more minutes because I think we have a lot of people more to join in. So, yeah, I think I want all of you to be a bit more patient. Great. I think it's the time for us. So, hi everyone, and good evening to one and all. I hope all of you are staying safe in this COVID pandemic and studying as well. So, I'd start by introducing a little about myself. So, hi everyone, I'm Bhavya Bhava, and I'm the current and the founding president for Walter E. Dandy Neurosurgery Club. I welcome you all on
Like
behalf.
your microphone once, Bhavya.
Am I not
Properly.
audible? Oh,
Yeah, you're audible, but slightly.
yeah, I'm audible, right?
Okay, yeah.
Okay. Great. So I think we can do it all over again. And hi, everyone. So I'm Bhavya Bhava, the current and the founding president for Walter E. Dandy Neurosurgery Club India. And this is, a first of its own kind club, which is typically for a super specialty and that to neurosurgery. And I welcome you all today on behalf of my whole team. So we are here today and we welcome Dr. Anudeep Yekula, who is currently the general surgery preliminary resident at the Yale New Haven Hospital, Connecticut, US. And he's been a postdoctoral research fellow at the Massachusetts General Hospital, which is, our own MassGen. And that's associated with the Harvard Medical School, again, Boston, US. Like that's my dream college. I think every medical student's dream college it is. I'll begin by introducing Dr. Yekula. So he's graduated from Guntur Medical College, India, as a valedictorian with several honors and scholarships. After graduation, he joined the laboratory of Dr. Bob Carter, who is the current chairperson for neurosurgery at MassGen itself. His research has been focused on developing liquid biopsy assays for minimally invasive diagnosis and monitoring of brain tumors, especially glioblastoma. Understanding the biology of cancer is fundamental to developing meaningful therapies, and liquid biopsy offers a unique avenue to study the spatiotemporal heterogeneity of the tumors at the diagnosis and follow-up. Dr. Yekula's career objective is to be a surgeon scientist, combining clinical practice with research and discovery to innovate, improve, and provide the best surgical care to patients. I welcome you wholeheartedly, Dr. Yekula. Thanks for being a part of this session today, and we are, with warm regards, the stage is all yours. Thank you, sir.
Thank you so much for the kind introduction, and thank you for having me here. And I want to first thank the entire team for spending your time to be with me, to listen to my talk, and special thanks to the organizers, especially Salekia, for organizing this and having me on board. So I can start my presentation. I'll share my screen now. Okay. I hope you're seeing my presentation screen right now. Are you?
Yes, sir. Yes, sir.
Okay. So this is Anudeep Yekula. Like the organizers have introduced me, I graduated from Good Medical College and decided to pursue my residency in the United States. And I worked with Dr. Bob Carter at Mass General Hospital for about 2.5 years. And my primary research was focused on developing blood tests for the diagnosis of brain tumors. And I'm very happy to share my work with you all. And these are my funding sources and disclosures. So how I want to structure this talk is, first, let's discuss the challenges in diagnosing and monitoring brain tumors, because it's a little different when you compare it to other solid tumors or other tumors. And then we'll try to understand what liquid biopsy is and how liquid biopsy can address some of these challenges. And then I'll give you a little bit of overview of some of my projects in the context of liquid biopsy and brain tumors. And then we'll discuss the path through research in the US or otherwise. So in the paradigm of management of glioblastoma, we know that glioblastoma is the most aggressive type of glioma, or most aggressive brain tumor. It's an extremely challenging and a complex disease. So patients usually present with headache or focal neurological deficits, and then you go take them to an imaging center and get them a CT or an MRI. Imaging provides an indication of tumor presence or the tumor location, but it cannot provide you the details on histological or molecular diagnosis. So you want to know what the molecular nature of the tumor is. So the patient goes through a highly invasive brain tumor surgery for diagnosis or for gross total resection when possible. Later, they undergo chemoradiation and the patient is then monitored by serial imaging. The tumor eventually recurs because glioblastoma is extremely aggressive, but in very, very, very rare cases, it remains stable with a mean survival of about 6 months. And at recurrence, there is no standard of care. And in the context of brain tumors, especially from a diagnostic standpoint, surgery is highly invasive. Nobody wants to get a brain surgery to know what the tumor is. And biopsy can result in potentially life-threatening complications because a small nick to a vessel or small nick to a functional region can have devastating complications. And biopsy of tumors that are close to functional locations or difficult to access locations can be challenging, and such surgeries or biopsy procedures can result in permanent neurological deficits. And the tumor itself is highly heterogeneous. If you take a glioblastoma and break it off into 7 or 8, 9, 10 small pieces, you would notice that every small piece has its own histological and molecular characteristics. So it's highly heterogeneous. and a single biopsy cannot always provide the entire picture of the tumor. And repeated sampling is not possible over the course of treatment. And the tumor constantly evolves over time and therapy. And imaging-based monitoring cannot provide insights into the evolving molecular landscape. And these problems are not just unique to brain tumors, but to a lot of cancers. And moving ahead, what if we can diagnose a brain tumor at a molecular level, irrespective of its location, using a simple blood test? What if we can stratify patients to appropriate therapies based on a simple blood test? What if we can assess response to therapy using a simple blood test? And what if we can monitor for recurrence using a simple blood test? And thus, liquid biopsy can be the solution. Liquid biopsy involves the analysis of biofluids such as blood, CSF, urine to determine the characteristics of a tumor. In simple, liquid biopsy is a blood test for cancer. And there are three main areas of interest in liquid biopsy: the circulating tumor cell fraction, the cell-free fraction, and extracellular vesicle fraction. I'll give you a brief overview of what these are and how we can use these modalities to understand what the primary tumor is. Circulating tumor cells are cancer cells that leave the primary tumor and enter circulation. And in the context of some cancers, these cells can see different locations and metastasize or remain in the circulation. And if you're able to isolate these cells and study the molecular characteristics of that cell, you might be able to profile the parent tumor, come with a few disadvantages. CTCs may be representative of that aggressive subclone of the cells that have the capability to detach, so they might not be representative of the entire tumor itself. And glioblastoma CTCs cannot survive for long in extracranial locations, and that is the reason why glioblastoma doesn't metastasize. And because it's a cell, it cannot cross through an intact blood-brain barrier, and it needs disruption of the blood-brain barrier for the cells to make its way into the bloodstream. And thus, you can only see CTCs in very advanced stages of the disease. But the point of cancer detection is you want to detect it early. And CTCs are extremely rare because bloodstream has a million other cells. So the concentration is about 1 cell per 10 power 9 blood cells. And it requires a large amount of fresh blood for you to analyze it. And of course, all these liquid biopsy platforms require advanced technologies for isolating and characterization. Those are the disadvantages of CTCs. Moving on to circulating tumor DNA. Circulating tumor DNA is a cell-free DNA that's released by the dying tumor cells into the circulation. And as we know, the DNA is extremely stable. So studying tumor-derived characteristics based on circulating tumor DNA in the blood can give us insights into the molecular status and mutational status of the tumor. For example, if the tumor has a particular mutation that you're interested in, you might be able to take a sample of blood and identify that mutation. If you want to look at the methylation pattern of the tumor, you might be able to study the cell-free DNA from plasma and identify what methylation pattern the tumor has. And lastly, extracellular vesicles. Extracellular vesicles are these small membrane-bound nanoparticles, 100 nanometer size. And the size of a cell is 10 microns. And these are 100 microns in comparison to the size of the cell. So these are the small membrane-bound vesicles that are released from the tumor. And they carry genetic and proteomic material when they bud off from the cell. And they get released into the biofluids. And these EVs act as intercellular communication devices. So it's more like molecular tweets. Cells communicate with each other through extracellular vesicles and mediate several functions such as growth, proliferation, immunosuppression, whatnot. And they're actively secreted, in contrast to circulating tumor DNA, which are released by a dying cell. and extracellular vesicles can cross the blood-brain barrier and are very stable in biofluids. However, the challenge is that not just the cancer cells, but every other cell of the body also releases extracellular vesicles, or EVs, which leads to a very heterogeneous population of EVs in blood. To put you into context, 1 ml of blood has almost 10 power 12 EVs. And of course, it requires advanced technologies for isolation and characterization. And my research has mostly focused on circulating tumor DNA and extracellular vesicles. And now I'll move on to a few of my topics or my projects based on the background we just built. The first project I want to talk to you about is the 5-ALA project. Like I told you, every cell of the body releases EVs and not just the cancer cells. But we are interested in that small subpopulation of EVs that are released by a cancer cell. And we want to look into those tumor-derived EVs and study the genetic materials derived from the tumor. And that leads to the classical problem, needle in a haystack, where's Waldo? Because in a biofluid, you have millions and millions of EVs derived from every cell of the body. How do we identify that subset of EVs that are tumor-specific? So I've been studying 5-ALA as a strategy for EV-based liquid biopsy for the past 2 years. 5-ALA is a very interesting drug. It's a drug that's used in fluorescence-guided surgery for malignant gliomas. The protocol is the patient drinks 5-ALA 2 to 4 hours prior to surgery. 5-ALA gets absorbed, reaches the brain, and gets absorbed into the tumor tissue. It gets metabolized into a fluorescent molecule called as protoporphyrin. However, in other cells of the body, 5-ALA goes through the heme synthesis pathway to become heme. But in cancer cells, due to disrupted metabolism, it stops at an intermediate molecule called as protoporphyrin. And when you shine blue light, the tumor fluoresces pink. This helps the neurosurgeon to identify the tumor based on the fluorescence and perform maximal resection. I'll show you a small video to put that into context. Here you can see the pink fluorescent tumor and the normal brain tissue, which is blue. So an operating neurosurgeon can look at the fluorescent tumor and resect the pink glowing portions and leave out the healthy brain tissue. So our central hypothesis for my research is, does a glowing tumor release glowing vesicles? If yes, then we can study these glowing fraction of vesicles to identify tumor-specific characteristics. So we resorted to this highly sensitive instrument called as imaging flow cytometry. And we've developed a novel gating strategy to study these fluorescent vesicles. A little bit about the technology, not too much. So we take cells in vitro, dose them with 5-ALA, and take conditioned media. Conditioned media is like the broth in which the cells grow. And EVs are released into that conditioned media. And we take that sample and we run it through this instrument. And there is a microfluidic system through which the cells, or the EVs in this context, flow one by one. And you shoot it with a light of your interest and look at the fluorescence. And because it's an imaging flow cytometer, it takes an imaging, or it takes a picture of a bright field image as well as the fluorescence. And that is how, I mean, it's a lot more complex, but that's the overall picture. And that's how we were able to identify these fluorescent vesicles. In our initial experiments, we dosed glioma cells with 5-ALA, and we identified that they release fluorescent EVs, while control cells, which are human brain endothelial cells or any other cell, for example, when we dose that cell with 5-ALA, we haven't seen any fluorescent vesicles. Again, for all intensive purposes, anything in this box are fluorescent vesicles due to the gating strategy that we developed. And later, we moved on to in vivo studies. And we showed that glioba-bearing mice dosed with 5-ALA also release fluorescent EVs. So we have 2 tumor mice, and we have dosed the mice with 5-ALA, taken blood samples prior to and 3 hours post-dosing. And we saw that there was a consistent increase in fluorescent EV levels in the tumor-bearing mouse. And this is the fluorescent tumor in those 2 tumor-bearing mouse. And then we moved on to patients. And we also showed here that GBM patients with fluorescent tumors release fluorescent EVs. We analyzed the plasma of 6 malignant glioma patients undergoing 5-ALA-based surgery. In all the 4 patients who had fluorescent tumors at the time of surgery, we saw a consistent raise in fluorescent EVs post-ALA dosing compared to pre-ALA baseline. Also, the fluorescent EV levels highly correlated with tumor volume, which means that we saw higher levels of fluorescent EVs in large fluorescent tumors. And we published this study like 2 years back. But it's not enough to just detect that there are glowing EVs in the blood. We want to isolate them. We want to go in and study what genetic material they contain. So the next challenge was we need to sort these fluorescent EVs. So we optimized this instrument called as Astrios Nanofax for sorting these fluorescent EVs. And here we've shown that glioma cells dosed with 5-ALA have fluorescent EVs, and we can sort it using this mechanism. A little bit about this technology. Similar to the other technology, we dose cells with 5-ALA, collect the conditioned media, and run it through this instrument. Here, again, you hit it with a laser of your interest and look at the fluorescence. And based on the fluorescence, there's a jet of air that just sweeps across the fluorescent EV population to separate them into this tube so that we have just a separated EV population of our interest. And extrapolating on that success, we wanted to know if these fluorescent EVs that we are isolating are actually tumor specific. How do we know that? We might just be isolating any subpopulation of EVs, right? So we had 1 patient in our cohort who had EGFR V3 amplified GBM. This is a specific, interesting mutation that's very common in the context of glioblastoma. And in our cohort of 8 patients, we had 1 patient with this particular mutation of interest. So what we did was we did this interesting experiment where we took 1 ml of plasma from this patient, and we also have taken 100,000 sorted EVs. We isolated RNA and we evaluated the genetic material using droplet digital PCR. And to our surprise, we saw that in sorted EVs, we have a very high copies of EGFR V3 mutation when compared to 1 ml of plasma or 1 ml of plasma from healthy controls, which goes to show that the fluorescent EVs that we are separating are in fact derived from the tumor. And putting things into perspective, here we have 100,000 EVs, but in plasma, we have 10 power 12 EVs. So we are drastically reducing the background so that we can pick up the mutations of interest. So then, moving ahead, we devised a cohort of 8 patients, and we have sorted fluorescent EVs from them and looked at the EV RNA and performed whole transcriptome sequencing to understand what types of RNA they have. And the recent results suggest that we have several interesting mRNAs, long non-coding RNAs and microRNAs, which are extremely important in the context of gliogenesis, cellular proliferation, growth, and other metabolic processes. And this is very latest data, and we're still analyzing to see how we can tie the genetic material and the fluorescent EVs to the parent tumor. We are expanding this technology into other cancers as well. So that is one of the projects that I've been working on and one of my primary projects. And moving on to other aspects of liquid biopsy, I want to introduce you to TERT project. This is another exciting project. We've all heard of telomerase, which is used to maintain the ends of the chromosomes so that cells can live on. What happens with aging is the activity of telomerase decreases and the length of the chromosomes keeps shrinking. But when there is a mutation in TERT promoter region, the enzyme becomes overactive and overexpressed. This confers immortality to the cells, and that's cancer. And TERT promoter mutations are extremely important in the context of several cancers: glioblastomas, oligodendrogliomas, bladder cancers. So we set on to detect this mutation in blood. So we use droplet digital PCR for this. Again, PCR, is where you amplify the genetic material, be it RNA or DNA, in a blood sample or in any sample. But digital droplet PCR is an advanced technology where you create these droplets. You take the sample and you partition it into several thousands of droplets. And then you amplify each individual droplet. And based on fluorescence, you read those droplets to say if that droplet contains the mutation of interest versus not. This is advantageous because you are partitioning the sample into thousands of small droplets and looking at each droplet individually. That helps you do absolute quantification, as opposed to traditional PCR, where you take the whole sample as one singular unit. So you only get the relative quantification of the nucleic acid of your interest, as opposed to droplet digital PCR. So we developed this TERT promoter mutation assay. So this is where the TERT promoter mutation is. So we developed primers. Primers, there's a forward primer and there's a reverse primer. Primers are used to amplify that sequence of interest. And we have designed wild type and mutant probe to detect the mutation of interest. And we combine this technology or this assay that we developed with a very excellent platform called Exsolution that allows us to take a sample of blood and isolate circulating tumor DNA. And combining with 7DG, I want you to know that TERT promoter mutation is a very high GC-rich mutational region. What happens when there is a lot of GC-rich region is that there are kinks and secondary structure formations in the DNA. And addition of 7DG will reduce this secondary structure formation, which helps to keep the DNA strand in the plasma straight so that these primers and probes can bind, amplify, and detect. And we tested this assay in a cohort of 157 patients from different institutions across the US. And we had 87 patients with TERT mutant gliomas, and we had 37 patients with TERT wild type gliomas and 34 healthy controls. And to our surprise, we saw that we were able to pick up this TERT promoter mutation in all those independent cohorts when you compare it to any other group. And we demonstrated a sensitivity of 77% and a specificity of 90% with a very high negative and a positive predictive value, which has never been done in the context of liquid biopsy before. And in addition, we also shown that TERT mutant allele frequency levels parallel the course of disease. In a patient with progressive disease, we saw that the TERT mutant allele frequency levels increased as the tumor came back, demonstrating that the TERT promoter mutation is derived from the tumor. And in patients where the disease remains stable, TERT mutant allele frequency levels remain stable post-surgery and over course of therapy. So this assay has tremendous clinical utility and can impact clinical decision making. Our assay is currently undergoing CLIA validation, and it might be available in hospitals in the coming next 3 to 4 years. And we are working on other interesting mutations in the context of brain tumors. And then moving on to another project. Again, I'm just giving overviews of these projects so that I introduce you to the world of liquid biopsy in brain tumors. This is an interesting project where we are asking the question of, can we use a sample of blood to stratify patients to a particular therapy? Which means, can we predict if a cancer patient would respond to a particular therapy or not? So there was a clinical trial that was going on in our institute, which was testing the efficacy of the drug dacomitinib in patients with recurrent EGFR amplified glioblastomas. In that group, only 8 patients responded to therapy, while 48 patients did not respond. So what we did was we took 7 responders from this group and 7 matched non-responders. And we asked, can we look at RNA from extracellular vesicles in the plasma and predict who would be a responder to a particular therapy? So we used exelution to isolate evRNA and did long RNA transcriptome sequencing to identify evRNA signature that can help us predict the responder to a particular therapy. Again, sequencing is also a little complex, but I try to simplify it here, where you have RNA, you make cDNA, and then break the cDNA down into small fragments. And you amplify those fragments, and you map it against the reference genome to identify what those RNAs are. So we did that. And then we saw that first, we can separate out healthy and GBM patients just based on plasma. So if you can give me a plasma sample, I can tell you if that patient has glioblastoma or not. And we see that all these genes, which are extremely important in the context of several hallmarks of cancer, for example, gliogenesis, cellular proliferation, invasion, apoptosis, metabolic remodeling, all these genes were highly expressed in the plasma of GBM patients, but they were quite normal in the plasma of healthy controls. And later, we saw that the plasma evRNA signature of responders, denoted by R, is quite different from non-responders. And specifically, we saw that these particular genes can help us predict who would be a responder versus who would not be a responder. Essentially, we can take a sample of blood and predict if a patient would be able to respond to a particular therapy. Imagine the implications of this. Just in this trial, we have 56 patients who underwent treatment with this drug, and there are 48 patients who did not respond to therapy. And that's where the field is heading, where you are trying to match the patient with the right therapy to improve survival. And liquid biopsy can also do that. So liquid biopsy is the future. And I think over the next 20 to 30 years, we'll have more blood tests, more applications, not just in the context of brain tumors, but in the context of any other solid tumor or a liquid tumor for that matter. And over the past 3 years, we were able to show that liquid biopsy can help you diagnose tumors, stratify patients to therapy, assess response to therapy, and monitor for recurrence. And thus, liquid biopsy opens novel diagnostic opportunities. And I think I just wanted to expose you to the field of liquid biopsy beyond the context of what we are used to studying in our regular medical curriculum. So I would like to thank my mentors and my team for supporting me through these endeavors. And we can take a small pause to answer questions and then move on to the research discussion, or we can continue. If anybody has any questions, I'll be happy to answer.
Sir, I'd suggest that we could take the questions as we transfer towards the, I think we do have a lot of questions for now. So the first
Okay.
question says that 5-ALA work only for glioma or for other tumors as well?
So 5-ALA works for a lot of cancers. Okay. But currently for clinical use, it's used in the context of gliomas. And we've tested this in several different cancers, and the efficacy or the way it works is a little different in the different cancers. So it means that the fluorescence is extremely high in certain cancers and low in certain cancers. So there are clinical trials that are being undertaken to use ALA for resection of tumors in the context of other cancers as well. So in simple, to answer your question, yes, it works in other cancers. And we're exploring this in other cancers as well.
Great, thank you. So the next question says,
Mhm.
does ALA diagnose only malignant cells or any sort of abnormal cells like benign tumor? Is it like a
No, it's
differentiating marker?
no, it's for malignant brain tumors. So benign tumors do not have that aggressive changes in their metabolic pathways that can lead to accumulation of protoporphyrin. So this is mostly useful for malignant gliomas. But you can see some fluorescence in the context of low-grade gliomas and meningiomas. But if you take a very benign tumor such as lipoma, I don't think it'll fluoresce.
Right. The next question says, can ALA test be positive for even dysplastic and damaged cells and give a false positive?
So with dysplastic cells, it doesn't do much. But again, there are some mucous membranes which have demonstrated some fluorescence. There will always be certain amount of fluorescence. But if you are looking in the context of fluorescence-guided surgery, you're in the brain. You don't care about other parts of the body fluorescing, right? So you have your field with a fluorescent tumor and you resect it. But when you look at the application in liquid biopsy, all these places where there is minute amount of fluorescence can act as a false positive. But when we are able to look at the genetic material, we can curtail some of those challenges because we can look at tumor-specific mutations or tumor-specific cargo in the fluorescent EVs. So yeah, that's a great question, but limitations exist there.
Awesome. The next question says, if EV also have genetic material, how to differentiate it from circulate tumor cells?
Circulating tumor cells are this big, EVs are this big. That's how you differentiate. So, I mean, cells are cells, right? And EVs are some subcellular particles. And cells are quite rare, like I was explaining, because especially in the context of GBM, not a lot of cells detach. Even if they detach, they cannot survive in the bloodstream. But EVs are stable, so they can survive longer. And it's the size that distinguishes. And again, the characteristics of a cell are quite different from EVs, which are acellular and small.
Okay, if EVs can cross blood-brain barrier, then how is glioblastoma not metastatic?
Because EVs are not cells. EVs are only means of communication. So, like I'm communicating with you, it's a message that EVs send to other cells or to other parts of the body, but they don't have the entire genomic material to go to another place and start regenerating. So it's just messages. And in one of our recent papers, we've like summarized this phenomenon where cells in the brain communicate with other cells to promote their own growth. So they have a small community where they communicate with each other, help their growth, proliferation, bring in more blood supply, and suppress the immune cells present in that microenvironment. I mean, what glioblastoma does is amazing, and EVs are very crucial mediators of why GBM is such aggressive tumor.
Great answer. How much cost-effective or liquid biopsies can they be used in India?
I mean, that's a great question, right? Like, you have to compare this to the cost of a surgery, right? It's expensive, but Do you really want to go through an invasive brain biopsy, stay in the hospital, have deficits due to the surgery itself, or go for an expensive blood test? Again, these are all under development, and it takes 20 to 30 years for it to come in the US or in the developed countries. And for us, it might take even longer. But the point is, having a blood test to diagnose brain tumors will allow rapid diagnosis. And of course, in the context of a country like ours where things are not as reliable, you can just ship a sample of blood to a lab that is capable of doing it and get your results. So I think it has more utility for a country where the resources are limited because of the factors that I just described.
Great, thank you. I think we have a last question for now that says, can TERT mutation be considered in context to other tumors as well?
Yes. There are several cancers like bladder cancer, oligodendrogliomas, that have high levels of TERT promoter mutation activation. So because we are a brain tumor lab, we've tested this in the context of brain tumors, but other groups have been testing liquid biopsy application for TERT in other cancers, and we want to expand and test other cancers as well. So yeah, it's relevant in the context of several cancers.
Thank you, sir. I hope all those who ask these questions, they have their doubts cleared. And I think, sir, at your convenience, we can start with the research experience part, or if you want to break, we can do that as well.
No, we can continue. Okay, let me share my screen again.
We have one more question, I think, which says
Yeah.
how to differentiate ALA if that's present in other metastatic cancers.
I mean, usually a single person has a single tumor, right? So unless you have glioma and a meningioma, usually it's not a problem. And also imaging and the clinical condition of a patient will give you an indication of what type of tumor it might be, because meningiomas are quite superficial and glioblastomas are quite extensive and within the glial substance. So usually a person has one brain tumor, so it's quite easy to understand which type of tumor it is.
Thank you, sir. I hope Raghav Lakshmi's got her answer. Sir, at your convenience, we can start anytime.
Mm-hmm. Okay. So you guys can see the screen again, right? Can you see my? Yeah. Okay. So let's keep this discussion like a little casual and try to walk through the decision making of, do I really want to do research? And if the answer to that is yes, how should I approach it? Okay. And I don't want to like restrict this discussion to people who are interested to go to the US. I just want to keep it broad. So again, research is not a very straightforward thing. It's not like, oh, let's do research. It's a journey. It's a path. It's a path of discovery, and it takes a lot of your time, effort, and you need to carefully make the decision of going about research. But I promise, research is amazing. Like you've seen what we are able to do. So the questions that I asked myself when I was going down this route is, why do I want to go to the US, or why do I want to be in India in certain area, or why do I want to go to Ames or whatever, right? Am I doing because there is advanced medicine there? Am I doing it because I have all my peers doing the same thing? Am I doing it because my parents want me to do that? So I think understanding what your primary goals are is extremely important. If you're really interested in advanced medicine or trying to understand health policy or trying to understand public health, that's when you have to choose research, because that'll give you opportunities to explore those avenues. But if you're going to the US just for a good lifestyle, you don't need to worry too much about this. So what I'm trying to say is set your priorities straight right up front. And then the second question is, which branch am I interested in, right? So if you want to go to the US and if you're interested in surgical specialties, you need to do research because I think it expands your mind both as a clinician and as a scientist. And because you're a clinician, you can understand the problems in the clinic and you can go to the lab to answer those questions. You can do research even if you want to go to medicine or neurology or anything else, but they are less demanding in terms of research requirements. But if you want to go into certain branches, but in premier institutes, again, the research will give you that extra boost. So just choose if you really want to head down the path of research based on the branch you want to go with, because certain branches require research. Because we are competing against the elite pool of students from top US colleges who have been doing research from their high school. So that is why we will have like good scores or decent scores, but we are more of unidimensional people and they are looking for people who are multidimensional. We got to do research, we got to have good scores, we have to demonstrate leadership. So those are things that you want to develop and portray as you go down the route and not just have good scores. And then the next question that I ask myself is, why do I want to do research? And here it's like, do I want to do research because I'm interested in basic science work? Am I interested in clinical decision making in the terms of which drug would function better? Am I interested in medical education? Am I interested in policy? You need to ask yourself why you want to do research. It's not, I mean, people who do research just to get into residency will stop doing research the moment they get into residency, but that's not how it works. They want you to continue that academic productivity and be more than just clinicians. They want you to advance the field of medicine. So if your goal is to do that, that's when you have to choose research as a path. And then the question is, what kind of research am I interested in? Research is not restricted to advanced genomics. Research is any question that you want to answer. And if you look at the situation around you, you can see that India have a lot of fundamental problems that have never been addressed. So research is not restricted to that genomics and advanced science. We have to answer a lot of questions, which means that there are a lot of scope for research. Even if you're a neurosurgeon, even if you're interested in neurosurgery, you can do research along the lines of how can you get students interested in neurosurgery? How can you so that's the field of medical education, right? So where you try to improve the medical education in our country and you compare it to the medical education in other countries, how they have exposure to these research opportunities or conferences or projects from a very young age, and how that allows them to be leaders in global neurosurgery and all. And then there is a field of this global surgery, global neurosurgery, global medicine, where you try to understand what are the discrepancies between healthcare in developing countries and developed countries. So that is global medicine. You can address such questions. And especially in the light of pandemic, you can see how the impact of a particular disease can be on a global scale. And then there could be clinical research where you test drug A versus drug B and see which is better. And you can do trials and you can do basic science like I'm doing. And you can do policy making where you can study how certain policies impact healthcare and what are the changes that needs to be done in the governance or in the decision-making process of the administration to allow the medical care to be delivered to people. And you can see around you that there is a lot of misinformation. How can you tackle that? So what I'm trying to say is, basically, you got to pick a question or a branch that you are interested in. And to know what that is and to know that path, it just takes one Google search. And then you can choose which branch you're interested in the terms of research, and pursue that. And then when you are able to get to these questions, and you have to choose a question that is passionate to you. I wish I could answer all the questions I just spoke about, but you have to choose a question that you want to travel with for 2, 3, 5, 10 years of their lives. So people dedicate their lives to answer one question. So you got to choose that question carefully so that you pursue that with all of your heart. And there's no point in doing research that you're not interested in. It's just going to be a waste of your time and your mentor's time. So you answer all these questions, not to me, but to yourself, to see if you really want to do research. And once your answer is, yes, I want to do research, then it goes down to how you pursue research. And this is how I want to break down. And of course, we are in a very tricky situation. There are advanced medical colleges in India where there is top class research, but not everybody is from AIMS or JIPMER, right? Even I was from a very small medical college, but you have to seek opportunities to learn about research. I'm not talking about SPM textbook. I'm not talking about books. Research doesn't come from books because research didn't make its way into books. You have to learn about research through YouTube. YouTube has a lot of content in the terms of people presenting their work. If you're interested in glioblastomas, just Google or YouTube glioblastomas, and there are like hundreds of talks by the most top personalities in the field. Go learn, attend conferences, attend presentations like this, and you will learn about what everybody else are doing. And when you have that global exposure, then something will make you interested in certain aspects, and then you can pursue that. And then first, I would encourage you to ask opportunities from mentors at your home institutions. There will be someone in your home institution who is interested in doing small projects. You should try to find those people because our prior generation are not everybody is like accustomed to high-end research, but there are always few people who are doing research projects. It doesn't depend what project you are taking up in the first instance. Doing a research project will help you understand how to design a study. What are the challenges in designing a study? How to get the resources required to get that study? How to acquire data? How to address the deficiencies in data? How to do data analysis? And how to publish it? So you will understand the process of research, and the process of research is similar to any research. It doesn't matter which project you're on. The principles of doing a research remain the same. So don't find how to do research in textbooks. It's not there. So you have to do it from practical experience. And then you demonstrate that you have interest in research by taking projects from inception to completion. Don't just go to people, ask for research opportunities, and let that die off. Take that project with a complete passion. Learn every bit of it. Again, your mentors may only be able to show a little guidance, but you have to use sources to get more guidance. You have to self-tutor yourself so that you can take a project to completion. Be it a small case report, be it a meta-analysis, be it a clinical study. Make sure when you take a project, be the person who can take it to completion. And developing mindset. And I have heard from a lot of people here that research from India do not have a good quality. And I know why, because a lot of people try to take research that's done in the US and try to replicate it. But research is not about rewriting a paper. It's about creativity. So you should learn from what they're doing and make it your own and make changes and answer questions that are relevant in your context or in the context of your state or your place. And to do that, you need honesty, sincerity, and dedication. We don't want to be researchers who take somebody else's work, who just make up data when it is not there. And you need to be honest. and sincere to yourself and to the community. Because when you publish a data, when you publish a paper, it means that you're claiming things, you are saying that I identified these things. So it becomes a tremendous responsibility to put something out there and have dedication when you do this. Again, people in the US or anywhere, they're not looking for people who can score 270s. It doesn't matter. They're not looking for people who are genius. They're looking for people who are hardworking, sincere, and honest. Again, these are skill sets to design and research. You can learn how to do stuff, but you cannot learn to be honest and sincere. That's something that comes with you. So you have to develop these characteristics, and that's what they're looking for. You have to shed your egos and your things, and you need to be that humble person, people, because in the context of what people are doing outside, we are very small. And once you get that humility, you'll be